We good? Okay. Good evening. Good evening, folks. We're almost ready to start. Yeah, I just had to turn it on. Working game is fictitious. All right, folks, good evening and welcome. Very nice to have you here. My name is Tammy Richardson. I'm the chair of the Department of Biological Sciences in the College of Arts and Sciences, and I'm here representing the college tonight. I'm delighted to welcome you to tonight's Berger Lecture on cancer. The College of Arts and Sciences is a very richly diverse community of faculty and students. We're dedicated to the discovery of and dissemination and application of knowledge about the natural and human worlds, as well as the places they intersect. The college enriches the academic experiences of its students and is committed to impacting our community through faculty research, scholarship, creative activity, and public lectures like this one. It's really great to see a wide variety of faces and ages in the audience. We are home to a variety of academic departments, schools, and programs. We offer 38 undergraduate majors and 67 minors. We invite the community to participate in all the signature events of the College of Arts and Sciences. These offerings demonstrate the breadth and the depth of our scholarship. They're educational, celebratory, and fun, and I encourage you to sign up for the college's newsletter if you haven't done that already. Although the remarks don't say how you do that, but I will say see Morgan over here if you're interested. I'd like to take a moment to acknowledge Mr. Harris Berger. Harris, do you want to wave or stand up, please? Here he is, gentleman in the front, who together with his family established this lecture series in 2022 in honor of his brother, Dr. Franklin G. Berger, otherwise known as Frank. Frank, will you please wave or stand up? Or there he is. All right. Yay. And this honors Frank's hard work and dedication throughout a 35-year teaching and research career with the Department of Biological Sciences. Harris especially wanted to recognize Frank's passionate and tireless efforts in the establishment and the success of the Center for Colon Cancer Research and the Colorectal Cancer Prevention Network. Harris, thank you for your generosity to the University of South Carolina and for your vision in establishing this lecture in Frank's honor. This lecture series is intended to promote cooperation and the exchange of scientific knowledge and research that fosters further progress in the battle against cancer. In that tradition, we are thrilled to welcome Dr. Ashani Veeraratna from Johns Hopkins Bloomberg School of Public Health as our second speaker in the Berger Lecture series on cancer. I'd also like to give special thanks to the Berger Lecture Committee, Alyssa Armstrong, Philip Buckles, who's hiding way there at the back, Lauren Hofseth, I don't see where Lauren went, Sean Owens, Michael Wyatt, and last but not least, Committee Chair David Reisman. I'd also like to take a moment to thank the team who makes things run, the college's external relations team, especially Morgan Watson over here, Hannah Trumbull, and Ashley Green, who have worked behind the scenes to handle all the details, hopefully ensuring a wonderful visit for our speaker and a great experience for our guests here tonight. Now, I'm pleased to welcome my colleague, Dr. Alyssa Armstrong, to introduce tonight's speaker. Alyssa joined our department in 2016 after postdoctoral and postdoctoral studies at Johns Hopkins, and you'll see in a minute, she's got lots in common with our speaker tonight. Alyssa has established an exciting research program focused on understanding how nutrient sensing by multiple organs and tissues influences the activity of adult stem cells. Her research has important implications for our understanding of the association between obesity and increased risk of diabetes and cancer. So, Alyssa, please join me at the podium.
All right, thank you. And I have the distinct pleasure of being able to introduce our guest of honor tonight, Dr. Ashi Ratna. And first, I just want to kind of go through her educational history. She was an undergraduate student in biology at a small liberal arts college in Maryland, St. Mary's College. And I would like to point out that I learned today that her research experience, summer research experience there as an undergraduate student, really shaped her interest and strength and interest in research and has developed her into the wonderful researcher she is today. She did her doctoral work at George Washington University Medical Center in molecular and cellular oncology, and then her postdoctoral training at Johns Hopkins Oncology Center, where she assessed the mechanism of action of tyrosine kinase inhibitors on prostate cancer. She started her professional career at the NIH in the cancer genetics branch as a staff scientist, and then after a few years, earned a position as head of the cancer biology unit at the National Institute on Aging. In 2011, she transitioned to an assistant professorship at the Worcester Institute, where she was later named as the Ira Brand Associate and then Associate Professor and a Professor, which is an endowed professorship. In 2019, she made her way back to Hopkins and kind of associated with two aspects of Hopkins. First, at the School of Public Health, in the Department of Biochemistry and Molecular Biology, which is where I happened to do my postdoctoral training and ended a few years before she joined as the E.V. McCollum Chair of that department. And then she's also at the Sidney Kimmel Cancer Center, where she started off as co-lead of the Cancer Invasion and Metastasis Program, and is now also the Associate Director for Laboratory Research there. So her research interests, broadly speaking, have been focused on understanding the cellular and molecular underpinnings of how the tumor microenvironment influences cancer progression and response to therapies, with a particular interest on the role that aging plays in that. Her research has been quite successful, as evidenced by hundreds of publications, consistent and significant funding support, speaking engagements as keynote speaker at national and international conferences. And this also includes a book that she wrote called Is Cancer Inevitable by Johns Hopkins Wavelength Publishing. She has received several honors and awards. And as I looked over her CV, it looks like almost annually, when she was at the NIH, she received kind of a staff recognition award, this On Spot Awards. In 2022, she received the Paget Ewing Award, which is the highest honor, and I'm reading this from the website, the highest honor bestowed by the Metastasis Research Society and honors a person's scientific excellence and substantial contributions to the understanding and or control of cancer metastasis. And potentially most notably, she was appointed as one of the 7 clinicians and researchers to the National Cancer Advisory Board by former President Biden. Outside of her research accolades, she has utilized a variety of platforms, radio, podcast, TV, to communicate with the public about the importance of her research findings and research in general in promoting skin safety. And most importantly to me, she is a true advocate for supporting and encouraging women and people of color in science well before it was a thing to do. And she will continue to do that, with what I hear. And this can be seen in both her professional and public service activities, where she has organized a Women in Science Symposia pretty regularly for the Society for Melanoma Research, and has participated in outreach activities in middle and elementary schools in Baltimore. So on behalf of the Berger Cancer Lectureship Committee, I would like to welcome Dr. Weeraratne to the stage, and so that we can hear all of her lovely work and we can applaud. And while we are transitioning, I do want to make a note that all of you can see that there are note cards on the tables in front of you with pens. As you are listening to the lecture, if you have any questions that come up, please write those questions down on those note cards. And at some point, we will pass them down to the student ambassadors, where we will collect them, and Dr. Reisman will be facilitating the question and answer session. So thank you and welcome to the stage.
Thank you. All right. Do I need a microphone? No. Okay. All right. So first of all, thank you for that amazing introduction. It's the kind of introduction that I wish I could tape and send to my mom, because she would be very excited to hear that. So thank you so much. And thank you to all of you for the amazing hospitality that you've shown me. I've heard about Southern hospitality. It's my first chance to experience it, and it has been absolutely lovely. So thank you. Thank you to the Berger family for having me here. It really is an honor to be here. So I thought I'd start by telling you a little bit about my background. And I started out in this tiny little country over here. Oh, does this not work? Oh, there it is. It's very faint. I think the battery's dying. But anyway, I started out in this tiny little island over here called Sri Lanka, where I was born. And then when I was 1, we moved to Africa. I started out in Zambia, but I spent most of my life in Lesotho, which for all of you, trivia question, it is the only country that's completely landlocked by another single country. The Vatican tries to claim it, but we all know that's really Italy. When I was 17, I made the decision that I wanted to go into cancer research and into medicine and science in general. But at that time in South Africa, it was apartheid. And as a woman of color, I knew that I could not pursue my education successfully there. And so I made the decision to come to the United States for college, and I ended up at St. Mary's, as Alyssa pointed out. And now, after a journey basically up and down between Philadelphia and Maryland, I have ended up back at Johns Hopkins, where it has been a really lovely time. And I will say the one thing that all these 3 places that seem very disparate have in common is that they're all absolutely beautiful. Sri Lanka is islands and beaches, Lesotho is mountains, and Baltimore has its own charm as well, no matter what you've heard. It's an absolutely beautiful city. So please come visit us. So I thought I would put a couple of slides in, especially for the students in the room. So as you guys are starting out your careers, you kind of have an expectation of what a career path looks like. And you expect it to be a little uphill. But what you don't realize is that actually it's going to kind of look like this. And so there are going to be a lot of peaks. There are going to be a lot of valleys. There are going to be some sharks in the water. But I want you to look at one very particular thing on this slide, which is if you look closely, no matter how dire things, oh, sorry, I thought I had taken that out. This is just the career path. But if you look closely, no matter how dire things look, you will always find that there are little ladders, ropes, and bridges that can help you get out of those hard times. And so it's very important to, Mr. Rogers used to say, look for the helpers. That's also true in science. Look for the lifelines. And what are examples of some of those lifelines? So I have been very blessed and very lucky to have had along the way amazing mentors and sponsors. I still have mentors to this day, even at this stage of my career. And then in addition to the mentors and the sponsors, I have been very lucky to be blessed with wonderful friends, both in science and both outside of science, which is very important to have both kinds of friends. And I've also been incredibly blessed to have amazing trainees who are like family. My trainees really are my biggest source of support. And then, of course, there's family. I've been lucky to both have a core family as well as an adopted family, because when I moved to the United States, I wasn't 18 yet, so I had to have legal guardians who became my family over time. And then, of course, the people I depend on most are my little nuclear family, my daughter, my husband, and we lost the little fluffy one a year ago, but she still holds a big place in our hearts. So find your lifelines, find your support systems. And what I'll say is that it's so important to build a community for yourself, both within your lab, within your school, but also outside of it, because sometimes having that outside perspective is going to be so helpful for you as you move forward in your career. The other thing we'll talk about today is about following the science, even if it tells you you're wrong. I'm going to give you an example of that as I talk about the science in a minute. And then for the students in the room, not only should you have mentors, but you should also identify sponsors. And so a sponsor is someone who is not necessarily the person you go to and cry on their shoulder when your experiment doesn't work. It's the person who thinks of you when there's an opportunity for a talk or to write a review or some other way to further your career. And those people you meet a lot of times at meetings, or maybe your mentor will introduce you to them. So keep those people in mind and turn to them when you need them because they are going to be very important as well. Um, so yeah, I've had cause because of the work that I do in the diversity space to generate my fair share of haters as well. And so, at some point you've got to learn to separate the noise from the work. And you need to develop a little bit of a thick skin. But my sort of guiding star has always been if I am doing the right thing and if I am working with integrity towards a goal that I firmly believe is the right goal, then I have nothing to worry about. So, and Winfrey said it best, real integrity is doing the right thing, whether or not anyone knows you are. And so, and then my last point, so none of this is in order, I realize, but my last point is that, what has been really useful, I'm very much a list maker and a planner. And so I like to make myself strategic plans for different parts of my life. And so, and those plans change, obviously, as you go from career stage to career stage. And so when I took on the job of chair 5 years ago, I made myself a strategic plan. This was my strategic vision for the department and for myself. And that plan was to increase the inclusion in our department, increase the diversity within the department, and increase awareness and perform more community outreach activities. It was to work on capitalizing and expanding innovation, and it was to work on collaboration. So my department is a basic science department in a school of public health. And in a self-study that had been performed before I got there, one of the criticisms was the department is very siloed. And so one of the things I really wanted to do was to build bridges and increase collaborations between the faculty in my department and across the school and across the university. And then, of course, something that has always guided me is wanting to have an impact to help people and whether it's through our science or whether it's through an impact that we have in a societal setting. And so we'll talk about all of those things. So in terms of inclusion, it has been really important to me over time to write op-eds, to talk to people about the importance of creating a safe space for everyone in science, right? So we often say in my lab, it doesn't matter who you love, it doesn't matter what you look like, we're just here to do the science. And our goal is to train the best scientists to go out there and eventually find cures for cancer. These are some of the faculty I've had the good fortune to hire since joining Johns Hopkins. And I will say that these faculty all just rose to the top. Half of them are from Ivy League institutions like Penn and Harvard or HHMI and NCI. And we were also fortunate to recruit some Bloomberg Distinguished Professors to the department who are stars in their field. But of the junior faculty that we recruited, I just want to make the point, and this gets lost I think this gets lost in translation a lot of the time, but diversity doesn't mean compromising on excellence. These people were all the top of their field when I recruited them in. Many of them came in with grants. And in the few years they've been with me, they have, of the three that were hired 3 years ago, they all have federal funding. They've all sent out senior author publications. And of the two newest ones, they're all building great labs with great students. And I'm so proud of all of them. So that has been a lot of fun. Next is innovation. And so I will tell you a little bit about some of the science that we're doing, and then we'll talk about the other two as well. So one of the questions we've been asking in my lab is what conversations do tumor cells have with other cells around them? And how do those conversations change as we age? And why should we care about that last part? Well, it turns out that for every 100 people who have been diagnosed with cancer, almost 90% of them will be over the age of 50. And as you can imagine, with an aging population, that becomes a huge public health burden. So not only does incidence and mortality of cancer increase with age, the response to therapy changes with age, and in turn, therapy can also accelerate aging, which is a newer area that my lab is studying now. We often think about a tumor and how a tumor cell changes, but in my lab, we say no tumor is an island. It exists in this milieu of all these different cell types. And the question that we've been asking is, how do these normal cells change? And how does that drive changes in tumor cell behavior? So we have focused largely on fibroblasts. And what we find is that as they age, fibroblasts both secrete different factors and build different matrices. So the biophysical matrices that fibroblasts build are different when those fibroblasts are older. Many of the experiments that we do in the absence of tumor cells initially, and then we bring tumor cells into the mix. So we're not looking at cancer-associated fibroblasts, which is a question I often get asked. We're literally looking at fibroblasts that we've taken from either young or aged humans or animals. And what we find is that both the secreted and biophysical effects have impacts not only on the tumor cell, but on the endothelial cells, as well as the immune cells. So all of the different components of the tumor microenvironment. This is, again, just a slide to say that these are not CAFs. This is the most common question I'm asked. And what we find is that actually aged fibroblasts seem to be more primed to convert into cancer-associated fibroblasts, but they also exhibit multiple different features of cancer-associated fibroblasts, even in the absence of a tumor cell. So they're kind of already primed to be one, but they're not actually one yet. So over the years, we found that these aging fibroblasts affect multiple aspects of tumor progression, from therapy resistance to angiogenesis, metabolism. There are changes in the aged immune microenvironment. There are changes that drive metastasis, and there's also an impact on tumor dormancy. So when we first started these experiments, we were looking at dermal fibroblasts because we were looking at melanoma, which is a very aggressive cancer of the skin. And you can think of fibroblasts as the construction workers of the skin. They're the ones that build the scaffolds that cells sit upon. And as I mentioned earlier, they also secrete various different molecules that are important for cell growth in general, and also in tumors, very important for tumor cell growth. So what we have done is to take fibroblasts from normal healthy donors. So these people have no cancer. We take them from the upper inner arm so that it's not as sun exposed. And then we can use these fibroblasts to build artificial skin. So we build these artificial skins by putting in the fibroblasts, melanocytes, collagen, keratinocytes. And it's really kind of cool. We give them 18 days and then you can pick up the skin. It's kind of gross, but kind of cool out of a plate. And it looks like human skin, even under a microscope. And so when we do this and we put melanoma cells into this mix, the only thing that has changed here are the age of the fibroblasts that we put into the mix. So the melanoma cells are the same, everything else is the same. And in the settings with aged fibroblasts, you can see that the melanoma cells, which are these purple dots, are invading much more quickly into the dermis than in the young setting. And again, same cells, the only thing that's different is the age of the fibroblasts. We also show that in animal models, if we take a mouse, so this is a mouse model of melanoma called the BRAF P10 mouse. These tumors have been put into mice and then they're extracted and grown in culture. And now you can take those cells and put them back into an immunocompetent mouse, just young or age. And so that's what we do. We take a dish of these cells, split it in half, put half in a young mouse, half in an aged mouse, and ask what happens. We find that, interestingly, they actually grow much more slowly in the aged mice. However, they metastasize much more efficiently. So in the aged mouse, although the tumors are not very big, they are going to the lungs at a much higher rate. So they're forming greater metastases. So Mitch Fain, who's now an assistant professor at Fox Chase in Philadelphia, wanted to examine this. He wanted to know, was it that the tumor cells were going to the lungs at a faster rate in the aged mice, or were they going to the lungs and then growing out more quickly? So to measure this, what he did was to put tumors in young and aged mice, give them 3 weeks, look at the lungs for tumor cells, and then do the same over a time course. So I'm showing you here the 3-week and the 5-week points. And at 3 weeks, we found, so these little pink dots are the melanoma cells. They have M. cherry, that's what we stained for. So what we found is that in the young mice and the aged mice at 3 weeks, we would see these little tiny single cell colonies. If we waited just 2 more weeks and looked at the lungs of these mice, now we see that in the aged mice, these tumors have grown out really rapidly. But in the young mice, they remain in these tiny single cell colonies. So that's something we call tumor dormancy, where the tumors have left that primary site, they've gone to a metastatic site, but then they kind of sleep there for a while. And so our question was, what is changing in the aging lung that is driving these cells out of their dormancy, waking them up and making them grow? So Mitch did a proteomics analysis of young and aged lung fibroblasts, and he identified several different factors. Among them, there were some changes in Wnt signaling proteins. And so now students pay attention, because this is where Mitch did some work that turned a decade of thinking for me and my lab on its head. So when he saw these changes in the Wnt proteins, he did an analysis of all these different Wnt proteins in the tumor cells in the lung. And what he found was that in the young lung, the tumor cells expressed a ton of this protein called Wnt5a. But in the aged lung, where the tumor was growing out very rapidly, that's all the dark staining are tumor cells growing out. Again, same tumor cells, genetically identical, put in the just young or aged mice. He found that they no longer had Wnt5a. So that was really surprising to us because for years we had been saying that Wnt5a was associated with metastasis. All the papers that we published said Wnt5a equals invasion, Wnt5a equals, motility, et cetera, et cetera. And so when Mitch's data showed us that actually the tumors that didn't have Wnt5a were the big tumors in the lung, it really threw us. And so then we thought about it, and we thought about the experiments and publications that we'd had. And it turned out that what we had been looking at was invasion, so dissemination from that primary site, showing us that those tumor cells need Wnt5a to leave that primary site and go to the lung. But then once they got to the lung, if they still had Wnt5a, they would remain in this slow cycling dormant state. And to make those cells grow out, we needed to turn off Wnt5a. So it wasn't that high Wnt5a equals metastasis. Sorry, that was supposed to be crossed out. But this is the important point here is, Mitch was running all the right controls. And so always follow the science. Even if it tells you you were wrong, it led us to a more complex understanding of what metastasis is. So it's not just cells going to a different site. It's cells going to a different site, surviving there, and then growing out. And that very last step was a step that Mitch's work revealed for us. So just to prove this further, what he did was to make an inducible knockdown of Wnt5a in his tumor cells and an inducible overexpressor. So in the young lungs, he put in tumor cells where he induced knockdown of Wnt5a. And now when he does that, you can see that the tumor cells grow out very rapidly. What I don't know if you can see as well is that if he knocks them down very early, the tumor cells don't actually get to the lung. So that told us that we need Wnt5a for the tumor cells to go to the lung, but knocking it down made them grow out. He did the opposite experiment in aged mice where he treated the cells with, transfected the cells with an overexpressing Wnt5a, and he put the tumors in the aged mice. And now when he overexpresses Wnt5a, they remain in their little tiny colonies in the aged mouse lung. So what Mitch's work showed us, again, was a more complex view of metastasis than we had. We find that melanoma cells need to downregulate Wnt5a to grow in the skin. Then they upregulate Wnt5a, and when they do that, they can leave that primary tumor site and go to the lung. And then when they get to the lung, if they still express Wnt5a, they remain dormant. And then the aged fibroblasts change their secretory phenotype. They secrete molecules that inhibit Wnt5a. And that makes those tumor cells grow out and make these large metastases we're seeing in the aged animals. So this is just a summation of that, but basically what we learned from Mitch's work is that non-canonical Wnt signaling is critical for dissemination, but not for metastatic outgrowth. We identified this gene called SFRP1, and if we inhibit SFRP1, we can keep the tumors in these little tiny dormant states. So our current work is focused on immune regulation in the lung. What we have found is that in the aged lung microenvironment, there are a lot of myeloid-derived suppressor cells, and those are immunosuppressive cells. They keep the microenvironment in the lung immunosuppressive so that these tumors can grow out and grow out rapidly. And we're looking at ways to target those. This is also true in the brain. So it turns out that like lung metastases, melanoma also metastasizes to the brain. And in fact, it's one of about 5 tumors that metastasize to the brain. And it's when it metastasizes to the brain, it's largely incurable. And so we wanted to know whether there was an age association. And so I have a fellow in the lab who is a neuropathology fellow, and he went through the melanoma database, the melanoma patients we have at Johns Hopkins who had brain metastases. And when we look at the age of the patients with brain metastases, indeed, older patients have more brain metastases. So Cheyenne Palm in my lab, who is a PhD candidate, decided to tackle this project. And when she injects tumors into mice and gives them some time to metastasize and looks at their brain, she rarely finds metastases in young mouse brain. And, but she does find metastases in the brains of aged mice. So we wanted to know like what some of those factors were. And so Cheyenne is now in the process of doing that. And to do that, what she has done is to take brain, slice the brain into large, thick slices, and then take our melanoma cells and implant them on the brain. So it's an ex vivo slice experiment. This is what it looks like under the scope. This is a spheroid of melanoma cells that she has just implanted on top of a brain slice. And when she does that in young and aged mice, she sees such a striking difference between the way the tumor cells behave on the slices from young animals versus aged animals. And if you look here, these are the tumor cells. You can see that in the age, the tumors are migrating really rapidly, whereas the same tumor cells on a young brain slice don't metastasize as effectively. So that's some exciting new work. We're looking at the activation of astrocytes. It looks like the Wnt signaling pathway is playing a role there as well. And so we're pretty excited about that. So what I've shown you up to this point is that the aging microenvironment really affects things like metastasis. There are changes in the matrix, there are changes in angiogenesis, there are changes in metabolism of tumor cells, and all of this affects metastasis and therapy resistance. I'll tell you about that in just a second. And so my plea to everyone who does cancer research is to think about how aging is important. It's really important for us to consider the aging microenvironment as we're designing experiments, treating patients, etc. I'm going to switch gears for just a second to talk about intersectionality. This is one of my personal heroes. Her name is Kimberly Crenshaw. She's a civil rights lawyer, and she coined the phrase intersectional feminism, meaning that who you are is not just a single thing, right? You might be Black and a woman, or gay and Hispanic, or, an immigrant and white. But the way that who you are, all of those different things, not only affect the way you see the world, but also the way the world sees you. And so we talk about this stuff in lab meeting. And one of my trainees, who's now also an assistant professor, he said, well, we talk about this stuff as part of our society. But what about in our science? We already know age makes an impact. What about sex? And the reason he was interested specifically in sex is that when we look at the rates of mortality in melanoma, we know that men do worse than women. So the rates of mortality increase with age in both men and women, but men overall do worse than women. And so I put this slide up here just to say there are a bazillion factors that could be contributing to this, and we fully appreciate that. It's super complicated. But what Yash wanted to do was to break it down back to the fibroblasts and see if he could identify differences in male and female fibroblasts. So he did a ton of in vitro experiments, which I'm just going to brush over so I can show you the in vivo data instead. But basically what he showed was that aged male fibroblasts show more intrinsic aging than aged female fibroblasts. And one of the super interesting phenomenon that we saw was that young female fibroblasts actually senesced a lot more than either young or aged male fibroblasts. But that senescence was very different. So that senescence was not associated with what we call a senescence-associated secretory phenotype. And it was associated instead with the capability of cells to stop, repair their damage. So if we damage their DNA, the young female fibroblasts will repair their damage much quicker, whereas the aged male and aged female fibroblasts did not. So we think that this senescence was associated with repair and recovery, versus in the aged fibroblasts, that senescence is associated with a DNA damage response. We found that there were changes in metabolism in all of these fibroblasts, mitochondrial metabolism, cellular ROS, and all sorts of differences. But as I mentioned, I want to focus on the in vivo studies. So what Yash did, Yash was the postdoc he did so many, I don't know how he did the number of experiments he did. Like he's the only postdoc I've had to say to, listen, man, drill it back down again. It's too much. But so he did this massive experiment where he took young and aged male and female mice, and then he took melanoma cells that were male and female because we wanted to see, did the sex of the melanoma cells affect anything? And so he did this big crisscross experiment where he put the male cells in the male and female mice and the female cells in the male and female mice, and then asked what happened. So what we see is this. So in the male melanoma cells or the female melanoma cells, regardless of the sex of the melanoma cells, we see that they grow more slowly in the aged male mice. We see that they metastasize more effectively in the aged male mice. And we see that the markers of damage and cellular ROS and all of that, again, although we see an increase in aged females, are more pronounced in aged male mice. So we see a loss of base excision repair endonucleases, but then an accumulation of DNA damage markers in the aged male mice specifically. Again, no matter the sex of the tumor, whether the tumors are male or female. In melanoma, we actually have some great therapies. We have immunotherapy, as you all probably know. We also have really good targeted therapies. This is the oncogene, the BRAF oncogene, that is mutated in a large amount of melanomas. And we have targets that target this pathway very specifically. I'm sure some of you may have seen this slide before. This is a patient with melanoma. You can see that these are metastases all over his body. They're also in his lungs, his liver, his spleen. But when he receives an inhibitor against this gene, those tumors basically melt away. But unfortunately, after some time, they come back. So this is kind of old data. Now these patients lived 3-4 years after being treated with the BRAF MEK inhibitors. And what we had shown previously is that older patients have a far lower response to these BRAF inhibitors than younger patients. And that was really, really kind of startling to us because you have a mutant protein and a drug that targets that protein. And we thought nothing else should matter, right? You have such a specific interaction. But it turns out that when you put these tumors, the same genetically identical tumors, in young or aged mice and you treat them with this drug, they behave completely differently. So Yash wanted to know if that was the same with sex as well. So these are spheroid assays, and basically to drill it down, I'll show you the quantification in a minute, but to drill it down, the more red you see, the more cells are responding to the drug, the more cells are dying. So if we take these melanoma cells and we bathe them in conditioned media from the fibroblasts, if we bathe them in conditioned media from aged male fibroblasts, you can see that their response to the drug almost goes away. And again, in the females, it also goes down, but it's less dramatic than in the males, and that's quantitated over here. So Yash wanted to take this in vivo, and what he saw was really quite interesting because what he saw was what I told you before. When we treat young mice with a drug, they respond to the drug and then eventually they come back, just like in humans. If we treat aged mice with the drug, they think about responding, but then they don't really. They just keep growing. So those tumors just keep growing in those aged mice. In the females, though, it was completely different. In both the young and aged females, those tumors responded beautifully to the drug, and then it was only the aged females that started to recur after a period of time. So that tells us that something in the female microenvironment or the aged male microenvironment is driving resistance. Something in the aged male microenvironment is driving resistance to these drugs. So Yash, of course, being Yash, wanted to know what that was. And so he continued to do some proteomics, and he identified this factor, BMP2, which he found to be increased in the aged male microenvironment, whether we looked in the fibroblast, in tumors in patients, in tumors in mice, and when he treats with an inhibitor to the to the BMP gene, he can increase response to BRAF MEK inhibitors in these mice. The next thing he did was to see if he overexpressed BMP2, could he change the way the tumors responded in different microenvironments? So when he overexpresses BMP2 in aged male mice, nothing happens. They respond exactly as they did before. But now if he overexpresses BMP2 in tumors that we put into young male, young female, or aged female mice, now they really start to develop resistance to these drugs. So overall, what Yasha's work showed us was that dermal fibroblasts show both age and sex-specific changes, and those changes drive changes in the way the tumor cells respond in those environments. And he also showed us that male fibroblasts age faster, they have a higher rate of intrinsic aging, and that some of this is driven by epigenetic changes that drive the expression of BMP2. All right. And so, the other thing that I wanted to do when I got to Hopkins was to increase interaction in my department. And I like to lead by example. And so I collaborated with Dr. Liz Jaffe, who's our Deputy Cancer Center Director in the School of Medicine. And we co-mentored a fellow, Dan Zabransky, who is now an assistant professor as well at Hopkins. And Dan was interested in taking our aging microenvironment work and applying it to pancreatic cancer. And so what Dan found was that using pancreatic cancer models, if he injects tumors into the pancreas of young or aged mice, the same tumor cells again, they grow out more rapidly in the aged mice, and they also metastasize more efficiently in the aged mice. So just like melanoma, the aged microenvironment drives an increase in the aggression of those tumors. He wanted to know what it was that was driving that change. And he decided to, he was also influenced by his microenvironment. So he decided to look at fibroblasts. And we were lucky to collaborate with investigators at the National Institute on Aging who were taking pancreas from normal donors, deceased donors, obviously, who had no known pancreatic disease. And they kept the islet cells, but they threw out the fibroblasts. And so one man's trash is another man's treasure. And so we said, please give us your fibroblasts. And they were very happy to oblige. And so Dan used those fibroblasts to look at how they affected tumor cells. He found that these aged fibroblasts drove increases in tumor cell migration. These are just two different ways to measure migration. And he identified this protein called GDF15, which is a super interesting protein because it's also involved with cachexia, which is the muscle wasting that cancer patients get. And he found that aged pancreatic fibroblasts secreted a ton of GDF15. When we looked in the secretome of our fibroblasts, we found them. We found GDF15 in the tumors, specifically in our aged mouse pancreatic tumors. And we found that it was increased in patients as well, in aged patients. Dan then wanted to investigate the mechanism of GDF15. And what he found is that GDF15 activates AKT. And so these are just some experiments where he injected mice with recombinant GDF15, and he sees that there's an increase in the activation of AKT. If he knocks GDF15 out in his mice, so it's a systemic knockout, He sees that he can reduce the activation of AKT in these mice. And if he uses an inhibitor against AKT, he can reduce, this is just to show the inhibitor works, it reduces phospho-AKT. But what was really interesting is that when Dan does this experiment in mice that have pancreatic cancer, either young or aged, he's able to very specifically decrease the growth of these tumors in aged mice, but not really affect them in young mice. And so, and this is just to show that AKT is knocked down. So recently, Dan was able to get his hands on an inhibitor against GDF15 itself. This inhibitor is already being tested in clinical trials. And so what's super exciting is for the first time, I am embarking with Dan on a clinical trial. We're filing the paperwork, and we are going to treat pancreatic cancer patients with GDF15. So that is super exciting. And that is kind of, one arm of having an impact, right? Taking your discoveries, translating them into a clinical trial. But I would argue that in addition to that, it's also really important to have impact in other ways. So you heard Alyssa say that I wrote this book for the lay public called Is Cancer Inevitable? I'm always on social media being like the really unpopular person, reminding everyone as they head to the beach for Memorial Day to wear their sunscreen. And I have a funny story, which is when my daughter was little, and she was in kindergarten, all the kids, whenever I showed up for a sporting event, all the kids would run up and they'd throw their arms out like this, and I'd rub sunscreen all over them. And one time I got an angry call from the mom of one of Alina's, my daughter's friends, and she said, I'm at the pool and I forgot the sunblock and the kids won't get out of the car and I blame you. And I was like, yes, my work here is done. And so because I'm always talking about this stuff on social media, this guy reached out to me several years ago and he said, I've invented a dispenser, like, the alcohol dispensers to wash your hands for sunscreen. And I said, great, we're having a melanoma awareness day at the Phillies. Can you bring me a few of your dispensers and we'll debut them? So we did. For anyone who knows baseball, there's a famous baseball player called Mike Schmidt, who was a Philly. He was at the game that day commenting on the game. He saw these sunscreen dispensers. I only know him because I know nothing about baseball. I only know him because he's a melanoma survivor. And so he saw these and he thought that was great. He partnered with a foundation, and now these have been installed all over the Schuylkill pathway in Philly. So there's free sunscreen at parks and along the running paths in Philly. So that was exciting. We're trying to establish something like this in Baltimore. We kicked it off with a day at the Orioles where we handed out sunscreen and dispensers and talked about sun awareness. We had over 1,000 giveaways and we gave them all away. Now, I'm not going to lie, half of the people were really drunk, but I'm hoping they looked at the stuff afterwards. So the other thing, another great way to make an impact is to just be kind to everyone around you. It is very easy in a field where we are critical of each other and each other's science, because that's what we're trained to do, to lose that. And I think especially in times like the ones we're going through, I encourage you guys to be kind to each other and to your trainees, to your leaders in times like this. But most importantly, to yourself. Make sure you take time for self-care and taking care of your mental health as well. So these are the amazing people who actually do the work. I'm very fortunate to have a lab full of brilliant scientists, incredible collaborators at Johns Hopkins across the country and across the world. And I'll end, as I always do, with a picture of my favorite aging project, which is my daughter, Alina, who is growing up way too fast. Every time I pop up these pictures, I'm like, oh God. And I'll end there, and I'm happy to take any questions. I hope I didn't run over.
Yeah, well, thanks very much for a really wonderful talk. And we'd like to try something a little bit different than just having people raise hands. We thought we would get questions and then I would pick and choose the ones I wanted. So if anybody does have any, please, write them out and pass them on.
Where?
What?
Where?
Ends of the.
Oh, yeah, I guess, yeah. Okay,
Student masters of kids.
so there,
Sounds like I'm on a TV show.
yeah. So I'd like to start, I have a couple questions, not necessarily related so much to the basic research, but more with the psychology and sociology of young people entering into the field. And you mentioned early on that how important mentors can be. And I know this from my experience. I've had some wonderful mentors. And it doesn't only relate to science. I mean, it's really in any aspect of life. So my question is, do you have any advice, particularly to young people and also maybe to us as mentors, strategies to use to kind of choose a mentor? I know people who are entering graduate school and they have a hard time identifying who might be a good mentor. So do you have any?
I do.
Yeah.
So, for grads, for people picking a lab, for example, I think it's so important to talk one-on-one with the people in the lab and to also ask the person who's going to be mentoring you what their mentoring style is like. So, for example, people who want to come to my lab, I'm very honest with them. I have a lot of administrative duties. I'm not going to be in the lab with you holding your hand, but what I have done is to create an incredibly supportive lab environment where their colleagues within the lab will be able to help them with experiments. I meet with all of my students every week and talk through their projects and troubleshoot, but I'm not going to be the person in the lab showing you how to type files, for example. And so I think it's important to set up those honest conversations. What do you expect from a mentor? What would you like to see from a mentor? And then if that mentor's style is not what you would expect or want to see, that's maybe not the lab for you. It's so important to talk to the people in the lab as a student going into the lab. I also use my lab when I recruit people into the lab. If my lab says to me, that person doesn't fit, they're not a good citizen, or they need more hands-on than you can provide, or whatever it is, right? It could be anything. Then if my lab tells me they're not the person for my lab, then I respect them because they're the ones who work with them on a day-to-day.
So it's kind of a takeoff on that in a sense. Can you envision an ideal laboratory setting?
I think it depends on who you are, right?
Yeah.
So people who like to function independently, who want to go to a lab and explore their own ideas, then you probably want to go to a lab that is maybe more established, that has the bandwidth to follow more ideas that are not the PI's ideas. But then if you want to go into a lab where you're excited to get in on the ground on a project that's already running, that's a whole different ballgame, right? So I think it's such an individualized experience, and I think that's really important to recognize. There's not one good mentor. I might be a great mentor for some people, but just not be the right style of mentor for others.
Thanks. So are there some that will come up? Yeah, I got the tip. Who's here?
Frank has a question.
Okay, we'll take Frank's first.
He's selecting.
Yeah. Okay, here's a question from Dr. Berger. There's a lot of evidence that cancer rates are increasing in younger adults. Are aging fibroblasts involved in this? And are fibroblasts undergoing accelerated aging?
So thanks for that question. It's actually an ongoing project in the lab that a grad student has just started. What he's looking at is the signatures of aging colon fibroblasts and colon stroma to see if in early onset colon cancer, we see the same pattern. So are there signatures that are, prematurely aged in young people? And then, of course, the next logical step there would be, what is it that's prematurely aging them if those exist?
And another one here. You find that later passage fibroblasts also show changes, or is it only the age of the individual from the fibroblasts were derived from?
That's a great question. So we only use very early passage fibroblasts for specifically that reason. Because when you passage the fibroblasts for too long, they undergo senescence, and then senescence is a whole other ballgame. I will say that the secretome of aged fibroblasts doesn't always reflect a senescent secretory phenotype. But so we just use early passage fibroblasts because that's the question we're asking.
And even if they're isolated from patients of different ages.
That's-
Another one is, do you see aging signatures in the fibroblasts that are known to have been chronic exposure to any carcinogens?
So yes, and that's a great question. So when we first started this work, we noticed that there were some fibroblasts that were from young individuals that looked really old. And when we went back and we looked at the history, they were 99% from young females with a tanning history. And so we have a project in the lab that's looking at how the UV age secretome, the photoaging, mimics intrinsic aging. So yeah, I didn't talk about that.
Yes.
And also, as a public service announcement, when we started the UV experiments in the lab, we were trying to look for the right machine to induce enough DNA damage, et cetera, et cetera. We bought one from Biorad, we bought one from Fission, none of them worked properly. And then I said to my student, you know what, let's buy one of those nail salon UV cross linkers. So how many of you get gel manicures? Anybody in the audience? Yeah, don't do that. Because those are the machines we now use to induce DNA damage in our fibroblasts.
How about other carcinogens like smoking?
Yes,
Any-
for sure.
yeah.
And we know that
Yeah.
dermal fibroblasts are especially
Yeah. What about any difference in aging and melanoma between lighter or dark skinned individuals?
That's a great question. So it turns out that dark-skinned individuals get a very different melanoma than light-skinned individuals. So dark-skinned individuals get acral melanomas, which are on the soles of your feet, the palms of your hand, and in your nail beds. And so, if you're a person of color and you have a dark spot in your nail bed, it's probably just a bruise, but to be sure, if it's there, persists for a few weeks, it's a good idea to get it checked. Caucasian people can also get acromelanoma, of course, but it's a very different than the cutaneous melanoma that we study. And there is an age association.
What about the effect of sex? Is that describable or can it be ascribed to hormonal? Yeah.
That's probably the most common question I get with it. So we have looked at DHT and estrogen. We could not find reproducible. We have some of it in the supplementary data of that manuscript for DHT. But for the estrogen, it was very interesting because we couldn't find a reproducible effect of estrogens on the fibroblasts, which is kind of interesting because, there's pregnancy hormones, there's menopause, and you would think that would be more reproducible.
A few more. And do you have any information on the effect of diet on
We have
aging?
not done that.
No.
So we haven't done the calorie restriction. I mean, there are obviously plenty of people who've done wonderful experiments
Yeah.
with calorie restrictions, but we have not.
And then the last few, there's one here and there's another question that I have as well. Well, here's one that, how often does melanoma actually metastasize to the brain? It's very common.
About 30% of them do, so it's relatively common.
Yeah.
There was a study that showed recently that melanoma patients who pass away, many of them have metastases in their brain, even though it wasn't diagnosed when they were alive.
And the last few questions are really back to some social issues. And one of them is, what role do you see for advocacy as distinct from sponsorship and career development?
Yeah, I
So,
kind of think of them as
yeah,
both a little bit of the same thing. If someone's advocating for you, they're also, in a way, acting as a sponsor. Is that the question?
I think, I'm only reading them. I can't really interpret.
So hopefully I
Intention.
answered that.
Yeah.
You know, I mean, obviously, an advocate could be someone who maybe doesn't have the power or the position to sponsor you for a talk or whatever, but they could maybe talk to someone who does have that.
Yeah.
That's also incredibly important.
I'm going to close with one that I have that I think is on just about everybody's mind.
Don't ask me about indirect costs.
Yeah, that was the question. That was the question. So the question is, with all the changes and proposed changes going on, NIH funding and reduction of indirect costs, really it's a double question. How do you think this is going to impact the science that's done? But in a sense, more importantly, what advice do you have for young people going into science, like how to deal with this?
Yeah, so I think we're at a time where we're a little bit at a crossroads here. It depends on how all of this is going to shake out, right? So if you remember in the first Trump administration, there was a plan on the table to do a 10% and to make indirect costs 10%. And that got shelved. The cuts to NIH got shelved. And so my hope is that after some reflection, perhaps some congressional intervention, these cuts won't be as deep as they appear to be right now. So that's my message of hope. But it is a troubling time. There's no doubt about that. And I think what's troubling is the uncertainty of all of it. But I would say that right now, we don't know how this is going to shake out one way or the other. And the best thing to do is to focus on your work, because the work that you do is critical and it's important, and we need to keep doing it as long as we can.
Are there any other questions that you could entertain? Okay, if not, then I'd like to thank the speaker again.
Could I ask-
Oh, sorry. Good.
You're talking about the melanoma cells going to the brain,
Mhm.
and am I then understanding that the cancer cell passes the blood-brain barrier?
Yeah.
So is that telling you that the blood-brain barrier is changing
Yeah.
with age
Yes,
also?
I didn't show you those data, but they're very striking. When we look at the vascularity of the age brain compared to the young brain, it's dramatically different. And so we do think that that is compromised already, just as a function of aging in the absence of tumors.
Thank you.
You're welcome.
That's a depressing statement.
So
Yeah.
we'll say, older patients respond better to immunotherapy. So there's that.
Yay.
All right, well, I want to thank again. Oh, another question, one more. Yeah.
Yeah.
Treating pancreatic cancer, like, assuming you're gonna then try to apply it to other cancers.
Oh, so for this specific treatment, we have not yet done the preclinical experiments to see if it is important in other tumor types. So this one will be specific, but we suspect that it's going to be important in other GI cancers. Melanoma, we don't think the dermal fibroblasts are doing a whole lot with GDF15.
Yeah, that was kind of like the rest of my question. It was like, how would you then change your approach?
Yeah. I think, in melanoma, for example, we see that fibroblasts secrete ceramides, so lipids, that in the presence of those ceramides, the melanoma cells upregulate a receptor that allows them to absorb that fat and then allows them to resist therapy. And when we target that receptor, they become sensitive to therapy. So for melanoma, it might be something different. It might be that receptor that would to target, but we don't have a good inhibitor for that yet.
I think that, let's thank you again.